The old idea of systems biology was kind of to say, again, to put it crudely, right, we're going to figure out which molecules are interacting with each other, and we're going to draw these big networks, these big sort of circuit diagrams of this molecule speaks to that one, speaks to that one. And there was no spatial component in it. There was no structure in it at all. It was all a kind, a kind of conceptual idea. But we now know that space is absolutely crucial. I mean, things move around in the cell. For one thing, there are these compartments, but also, as I say, molecules seem all the time in our cells to form these kind of clusters, literally these blobs. They're kind of like dense liquid-like blobs where a certain number of molecules get together and have these kind of conversations. It's totally a spatial thing. What seems to be really important for which genes are being read and which ones are turned off is the three-dimensional structure of this tangled stuff called chromatin, which makes up our chromosomes, which is basically the DNA with proteins that kind of help to sort of bind it. This chromatin is unpacked by some enzymes so that bits of it are physically accessible to the enzymes that are going to read out the DNA instructions, and other bits can't be reached. And so there's a whole language of the physical structure, the three-dimensional structure of our chromosomes that is utterly central to the way our genes work, and that just isn't there when you think about it just in terms of a sequence. But it's not only space, it's also time. But how these things happen in time is really important. Just last week I wrote about a paper where it turned out that the fate of cells in the very early embryo, whether they become one type of cell or another type of cell, it was controlled, you could say controlled by a particular gene, but not by just whether that gene is switched on or off. It depended on how long the gene was being turned into protein 4. If you got a sudden pulse of it, then the cells became one type. If you got a sustained amount of it, they became another type. And we see this again and again, that there's this temporal aspect to it as well. So we really have to think about things happening in biology in space and time rather than in this abstract informational space. And there's information in those dimensions, in the spatial component and the temporal component. And that includes information that's coming from outside, from the whole environment of the organism, but also from the other cells around. So that, whether one gene gets switched on at a particular point in development depends on what the other cells around that one are like and what they're doing and which signals they send. So we can only understand that process by looking at the whole structure, rather than just some self-contained program within an individual cell.